Good morning, everyone. Hope you all are doing well and staying safe. Today, I'm going to, as I mentioned earlier, I'm going to talk about viruses, especially the SARS CoV-2, that the pandemic we're dealing with right now. And then I will be back to our normal topics. So our next topic will be differentiation and development. So for Monday, I will be back to our main topics. But today, as I mentioned earlier, I would like to talk about this SARS-CoV-2 virus, because this kind of what we have talked about, the replication, transcription, and RNA and the regulation of gene expression. This all kind of brings it together, and we will talk about it today. So let's talk about just in general about the viruses first. So viruses, what are viruses? They do not belong to any kingdom. They are neither plants, nor animals, nor fungi, protist, or bacteria. So what is a virus? And you all know this already, I'm pretty sure, that virus is it's a submicroscopic infectious agent. It can replicate only inside the living organisms. And that's the reason viruses are not considered as living organisms, because they require a host cell in order to replicate. They cannot do that on their own. So when we talk about viruses, we don't really talk about dead or alive, because viruses are, you would rather say, active or inactive. So if it's active, it's inside a host. If it's inactive, it's outside a host when it's not doing anything. It's just mostly just kind of being there. Viruses can infect all life forms. As we have already talked multiple times, and a lot of work we talked about is in bacteria. And bacteria get infected with bacteriophages, which are viruses. So phages are usually DNA viruses, but most human infecting viruses are they could be both DNA viruses and RNA viruses. The one that we will be talking about today, the SARS-CoV-2, is an RNA virus. Whereas most, as I said, bacteriophages, the viruses that infect bacteria, are mostly DNA viruses, so they're a little different. Not all viruses go through lytic and lysogenic phase either. There are very few of these phage that integrate into the bacterial genome. Most other viruses that infect us, they do not integrate themselves into the genome. There are some exceptions. HIV does integrate itself, HIV virus, but these majority of eukaryotic viruses do not integrate. So when they're not inside, when the virus is not inside inside a cell, or it's not in the process of infecting a cell, then the virus exists in a form of an independent particle, and this independent particle is called a virion. This independent particle virion, once it's infect, we also call it a virion. So when these are getting ready to be released from the cell. So a virion consists of genetic material that could be DNA or RNA, depending on the virus, whether it's a DNA virus or an RNA virus. A protein coat, that will be the capsid, which surrounds and protects this DNA or RNA. And in some cases, including the one that we will be talking about today, there's an outside envelope. It's a lipid envelope that is a part of this virion. Some biologists consider viruses as life form because they carry genetic material, they can reproduce, they can evolve through natural selection, but most will say these are the organisms at the edge of life, so they are neither alive nor dead. So an individual complete virus particle is a virion. Okay? A single infection in an organism consists of millions of these virions. So when we say that this person is infected, or this organism is infected, that means there are multiple copies, multiple virions of this virus are present. Most infections also have variants of the virus. So that means within an individual, these viral genome can differ. That means they can have a little bit of change, a few nucleotides changes from each other, and that's called variants of the virus. And this is a few of you who worked on this paper on SARS-CoV-2, that they were looking at these different variants of the genome. Whereas when we talk about polymorphism, that's a little different term now. Polymorphism is presence of two or more clearly different forms of an organism. Here in this example will be viruses. So these are referred as alternative phenotypes. And when we are talking about polymorphism, we are talking about a population, not an individual. So there can be variants within an individual, but they're polymorphism. So different, you can think it as a strain, different strain of viruses in a population. So different viral strains within a population will be an example of polymorphism. Here it shows an immature versus a mature virus or virion. And these they have, so in this example there is an envelope, there's a nucleocapsid. All these, this is genetic material, is kind of fragmented at this point, reverse transcriptase, integrase, etc. Then mature, this is all together. All of these are tightly packed because this this genetic material will be inserted into the host cell, will get inside the host cell. Viruses occur in many different shapes. We talked about bacteriophages, so these are the complex structures. Influenza virus, that is related to SARS-CoV-2 that we will be talking about. These are spherical, round-shaped virus. This can be helical, this can be polyhedral. So here it's the example of some of the viruses here. This is papilloma virus, rotavirus, hepatitis C virus, influenza virus. All of these are spherical virus. Ebola virus is more of a it has a very different shape. Adenovirus is one of these polyhedral viruses. So these can be virus can come in many different shapes. Human coronaviruses. So coronaviruses, the name coronavirus come from the Latin word corona, which means crown, because the outer structure, when you look through the electron microscope, it looks like a crown. That's why they are named coronaviruses. There are 4 subgroups of coronaviruses: alpha, beta, gamma, and delta. There are total 7 human coronaviruses that have been identified so far. So the novel coronavirus COVID-19 is the 7th coronavirus identified in humans. The most commonly occurring human coronaviruses are 229E NL63. These are alpha subgroup viruses, and OC43, which is a flu virus, HKU1, and the flu and HKU1. So they are all flu viruses. So all of these have very similar symptoms. They all cause common cold, flu like symptoms, and these are commonly present in the population. So most everybody has been infected by these 4 coronaviruses. The less commonly present coronaviruses are these 3. So total these make 7. MERS-CoV, which is another beta coronavirus. It causes Middle East Respiratory Syndrome, or MERS. SARS-CoV, there's also beta coronavirus, is called the Severe Acute Respiratory Syndrome, or SARS. And SARS-CoV-2, the new novel coronavirus, which causes COVID-19. So all 7 of these are coronaviruses that have been identified in humans, but there are more coronaviruses in animals. So these coronaviruses, most of these are zoonotic viruses. These came to humans from animals. This OC43, 229E was the first coronavirus to be identified in humans in assuming, I think it was in 50s or 60s. And since then there have been a lot of different coronaviruses. 6 more coronaviruses have been identified. So now let's move on to SARS-CoV-2, the coronavirus that causes COVID-19, the one that the pandemic we are dealing with right now. SARS-CoV-2 is the name of the pathogen, which is a severe acute respiratory syndrome coronavirus 2, because the coronavirus 1 is already given to the to SARS virus. So SARS CoV-2 is the pathogen. It causes, the disease that it causes is called coronavirus disease 2019, COVID-19. So there aren't 18 coronaviruses, there are only 7. 19 means it's 2019, the year when it was identified. So COVID-19 is an acute respiratory illness that we all know now. These all of these coronaviruses are positive sense single-stranded RNA viruses, so positive single-strand RNA viruses. These coronaviruses are capable of causing illnesses that range from common cold for the 4 most commonly occurring coronaviruses to more severe disease such as MERS, where the fatality rate was about 34%. Like the SARS, the original SARS-related coronavirus strain that was implicated in 2003 SARS outbreak, SARS-CoV-2 is a member of beta coronavirus or beta CoV subgroup that I already mentioned that. Now, how do we know that this SARS-CoV-2 is a beta coronavirus or it's related to SARS? So this is really now amazing that soon after the first case was identified, or first few cases were identified in China, the genome was sequenced. So the genome of SARS-CoV-2 is 30,000 base pair long. And this genome, now there are thousands of SARS-CoV-2 genomes have been sequenced. So these very first genomes, when the scientists put these together with other genomes, other viral genomes, they found that it matches very well with the bat CoV REG13, which is a beta coronavirus genome. So these are all novel coronavirus genomes. So they have at that point that 5 genomes sequenced, and these all 5 genomes were very similar to each other, all came from the same place, Wuhan, China, and they all matched pretty well with bat CoV REG13. So that suggested or showed or supported that these are beta coronaviruses. It's a novel coronavirus that we haven't seen so far. Here, this is a little better representation here. If you look at this chart here, this is the blue line is this the bat CoV REG13. So this is the coronavirus from a bat that's matches pretty well with this coronavirus, this novel coronavirus. Okay, so these others don't match as well as this. However, even this, it's not a perfect match. It's not 100% match. It's about 94% match, because here you will see that it starts to dip a bit. 100% match will be right here. So it's a very, very good match. Now, so when we say novel coronavirus, what does that really mean? It doesn't mean that it's a new virus species. This virus has been in animals, but in humans it's new. So it's new to humans, not to animals. It's a zoonotic virus, like a lot of other pathogens. These spill over from animals to humans. The genome structure here, if you look at this, these are the 2 other coronaviruses, the 2 beta coronaviruses, SARS-CoV and MERS-CoV. And if you look at these, this is one of the things I also wanted to show you, as if you look at these genome organizations of these viruses, they look very similar to retrotransposons, the virus-like retrotransposons that we were talking about. So this is the ORF1, ORF2, and then there is this poly A tail. So very similar to the retrotransposon, virus-like retrotransposons. Here, this is the leader sequence that will have, that's from where it will replicate or transcribe itself. So if we look at the SARS-CoV transmission, since SARS-CoV, the original SARS virus was originated in bats, and it was spillover into the cats, and from it went to humans. MERS-CoV was a little different, because this is the Middle East respiratory syndrome, so it was also originated from bats, and then the intermediate host was these dromedary camels. From these, it went to humans. So, so this is very common for viruses to spill over from animals to humans. Now, if we think about SARS-CoV-2, where did the SARS-CoV-2 come from? So it looks like it might be originally came from bat, because it has a pretty good 96% identical to this Rhinolophus affinis, which is a bat, the genome that we just looked at here. So this is the genome of this RATG13, is the bat-cov RATG13. So it's quite similar, about 96% identical to SARS-CoV-2. So it's possible that it also came from the SARS-CoV-2 also came from bats. There's another study that shows that it might also have an origin in pangolins. So in pangolins, there's portion of the genome of this SARS-CoV-2 is very, very 99% similar to pangolin, the virus that's occur in pangolin. And this is, we will talk about this ACE2 receptor binding domain. That's a very important domain, and domain for these viruses, because that's allow them to enter into the cell. So this SARS-CoV-2 ACE2 receptor binding domain is 99% similar to a coronavirus isolated from pangolin, whereas it's only about 77% similar to this RATG13, the bat virus genome. So what this suggests is that there might be a recombination between these two viruses. So there might be the bat virus and pangolin virus might have recombined to make this new this virus that we are seeing now in the human population, this SARS-CoV-2. How the recombination happened, where it happened, that's still a question that's need to be answered. So it's coronavirus origins. Genome analysis suggests two viruses may have combined in a bat, a pangolin, or another wild species. So we don't know where, whether it's happened in a cave or in a forest. That's still under investigation. So one thing to keep in mind, that recombination in viruses is not unusual, and viruses can recombine if they're present in the same organism. But then the question here is whether the pangolin, the virus infected bat, or vice versa, or there was another intermediate host that we still don't know. Okay. So now let's look at the SARS-CoV-2 structure. The SARS-CoV-2 virion is about 50 to 200 nanometer in diameter. So this is SARS-CoV-2 structure. Like other coronaviruses, SARS-CoV-2 also has 4 structural proteins, and these structural proteins are spike, the S that stands for spike, E protein, that means envelope protein, M protein or membrane protein, and N or nucleocapsid protein. They have this RNA genome, and this RNA genome, the N protein, this nucleocapsid protein, it holds the RNA genome. So this holds this RNA genome together. S, E, and M proteins, so they create this viral envelope. Okay, so 3 proteins are required for creating this viral envelope, and this 4th protein is required to keep the RNA together, the genome together. The spike protein is a glycoprotein. It's a very important protein, and you will be hearing a lot about this spike protein in the rest of the slides. This spike protein is responsible for allowing the virus to attach and fuse with the host cell membrane. So there's a specific receptor that it recognizes on host cell membrane that it binds to and goes inside the cell. So it's an endocytosis, this receptor mediated endocytosis. How does SARS-CoV-2 enter into the cell? As I mentioned, that there is this receptor that's the spike protein recognizes on the host cell membrane. This receptor is it's an ACE2 receptor or angiotensin converting enzyme 2 receptor. So here in this paper, these authors, they showed it very nicely with the crystal structure of C-terminal domain of SARS-CoV-2 spike protein complexing with the human ACE2 receptor. What it really means is that's similar to SARS CoV, the original SARS virus. SARS-CoV-2 also uses this ACE2 receptor as an entry point. The spike protein binds to ACE2 and allows it to come inside the cell, enter inside the cell. These authors also looked at the different monoclonal, polyclonal, and using different monoclonal and polyclonal antibodies against SARS-CoV-1, the original SARS-CoV receptor binding domain, and to see if these receptor binding domains are similar for both SARS-CoVs, so CoV-1 and 2. But they found that these receptor binding domains are quite different, but they're still able to bind to the same receptor on the cell. What that means is the antigenicity between SARS-CoV-1 and CoV-2 is different because they have different receptor binding domains. Okay. Here, so this is just a representation of how these SARS-CoV viruses, CoV-1, CoV-2, both of them, they enter into the cell using the cell membrane receptors and the division or the replication of the viral genome. So here, this is a virion. This is this virus. There's the viral RNA. These are spike proteins and the envelope. These spike proteins, they recognize these ACE2 receptors. They bind to these ACE2 receptors. Once they bind to these ACE2 receptors, I will show you in the next slide that there are proteases that helps these to bring this complex in. This complex comes in. Here, this virus enters into the cell body and releases its RNA. So these proteases also make these cut in the envelope so that the RNA can be released. Once this RNA is released, the RNA uses the host cell to create a new virus RNA and assemble new viral proteins. So because the virus cannot do that without a cell, so it hijacks the machinery of the cell in order to make the copies of its RNA. Also, keep in mind, we talked about these RNA-dependent RNA polymerases when we were talking about RNAi. That's humans don't have RDRPs or RNA-dependent RNA polymerase, but many eukaryotes do have as a mechanism of RNAi, where they will take these small interference RNAs that are cut by the dicer and then make them double-stranded, make them the long molecules again, so that can be cut again by dicer in order to increase the number of siRNAs. Here, this RNA-dependent RNA polymerase is very important because that's the polymerase for this virus. This RDRP is an RNA replicase. So it's what it does is, because it's an RNA-dependent RNA polymerase, not a DNA-dependent RNA polymerase, it does not require an intermediate. It does not require an RNA to convert into cDNA and then replicate. It can directly transcribe RNA from RNA. So it makes these new virus copies, and it also translates the other proteins, because the genome also code for those other proteins, the 3 other proteins that we talked about, the envelope protein, the membrane protein, and the spike protein. So it will make all these proteins. All these proteins will together be then assembled and packaged. So these are new virions, virus particles, and once it's assembled, it will be released from the cell. So it kills the cell pretty much to be released from the cell, right? So spike proteins are primed by these transmembrane protease serine 2 or TMPRSS2, and this priming is essential for entry of SARS-CoV-2. And these are the proteases that I was talking about. So this is, again, this is your SARS-CoV-2, SARS-CoV-1 virus. These are spike proteins here. Now they made this nice flowery structures. This is the RNA genome. here. This is the ACE2 receptor that they both recognize, although they have different binding sites, but they both recognize the same receptor, both virus, and they will bind to these ACE2 receptors. This binding of ACE2 receptor will need to be activated by these proteases. These are membrane-bound proteases. This is a membrane-bound enzyme receptor. This is membrane-bound protease, and then these together, these are required, and then this will go inside the cell. Here you can also see that the inhibition of this activation or inhibition of ACE2 or TMPRSS2 are both strategies to control this viral entry into the cell. So once the SARS-CoV-2 virion attaches to a target cell, attaches to this ACE2, the cell's TMPRSS2 will cut open the spike protein of the virus. It's cut open the spike protein along with this ACE2, and then from here, once it's go inside the cell, and this will then release the RNA into the cell, and then this virus genome will force the cell to produce multiple copies of viruses that will infect more cells in the organism. Another difference between SARS-CoV and SARS-CoV-2 is that SARS-CoV-2 requires or uses the multibasic cleavage site in the spike protein, and these are essential for infection in human cells. So what is the role of this unique multibasic multi-arginine, because arginine is a basic amino acid residue, cleavage sites in human SARS-CoV-2 cells? This is the question this paper of Menital Journal of Molecular Cell asked. Why the multibasic motif? So this multi-basic motif is not unusual. Other coronaviruses also use these. For example, these human CoVs, OC43, HKU1, MERS-CoV use this multi-basic cleavage site, but not SARS-CoV. So it's different from SARS-CoV in a way that this required this multi-basic motif. Okay. What the function, the proposed function of this multibasic site is that it's increased the specificity or increased the efficiency of virus for infection into human cells. So the punchline, as you can see, the monobasic site is sufficient for SARS-CoV-1, but SARS-CoV-2 is the spike protein requires this multibasic cleavage site. Probably is essential for its ability to enter human sites. It's also probably it makes it more infective than SARS-CoV-1. Okay. So next question that comes as the tropism of virus. So viruses display specificity in their host species and target cell type. So the virus will not infect all hosts, so they have specific hosts, right? So most of these coronaviruses, they're animal viruses, but they've spilled over to humans. So for example, here are some of the examples. The myxoma virus normally infects a rabbit, but it does not infect humans. So that's the host tropism for this virus. This tropism can be, it's the ability of virus to replicate in a particular cell type, tissue type, or host type. So different viruses will infect different cells in an individual, or different tissues in an individual. For example, influenza virus normally infects lungs but not brain. HIV normally infects macrophages but not neurons. So different viruses have different cellular tissue or host tropism. Because SARS-CoV-2 is an acute respiratory syndrome virus, so you would assume that the tropism for this will be that it infects lung tissues as well, similar to influenza. Okay, so the other group that studied this, Ziegler et al., it's the same group that we just talked about earlier as well. They looked at these the binding of spike protein of SARS-CoV-2 to cellular receptor of ACE2. Now, ACE2 is found in a lot of different tissues. ACE2 is present in lungs, in heart, in arteries, in intestine, and kidneys. So this ACE2 is present in a lot of different tissues, because the function of ACE2 is usually to keep the balance between you say the dilation and contraction. So these use different peptides. So this ACE2 is present in different tissues. So, but where this SARS-CoV-2 binds to, in which tissues SARS-CoV-2 bind to ACE2? So all these experiments here, they suggest that it's in the lungs. So, and again, it goes with, it supports the symptoms as well, because it's a respiratory syndrome. This virus causes respiratory issues. So the authors propose that this SARS-CoV-2 uses this ACE2 in lungs, pneumocytes in the lung. This is another group that also showed this here. They did the single cell RNA expression profiling of ACE2, and by doing that single cell RNA profiling, they used the single cells from multiple different tissues, and they found that this virus binds to these ACE2 receptors in alveolar cells. Type 2 alveolar cells. It's a small population concentrated in the lungs. So here, these are type 2 alveolar cells. That's where it binds. So they narrowed it down. Okay, in lungs, where in the lungs? In lungs, it binds to these particular cell types. Now this is very interesting. So now it gets interesting. This is another group of Sangnak et al. They published that SARS-CoV-2 is not only present, not only binds to these ACE2 receptors in lungs, but it also highly express the entry factors of the SARS-CoV-2 highly expressed in nasal epithelial cells as well, and these are associated with innate immune genes. That's the previous study also shows that with the interferons can increase the number of these ACE2 receptors and binding. So ACE2. So this was really important finding because a lot of these tests and it's been using nasal swabs for to get the viral load to test for the viral RNA. So it supported that nasal swabs will they yield higher viral load than throat swabs in symptomatic patients. So this is a good way to test. There was this also, there's a conflicting evidence for ACE2 and TMPRSS2 expression in the upper respiratory system. So conflicting because most all the papers, studies from the SARS and MERS, and now they all showed that the expression is highest in lungs, in alveolar tissue. But this study using, again, single cell RNA sequencing database, it showed that the upper respiratory system, the nasal area also has the high levels of these entry factors. So this potential tropism of SARS-CoV-2 and not only in the lungs but also in the nasal cells. But what they suggested that the virus might exploit secretory pathway in nasal cells at a presymptomatic stage. This is important for transmission, and this is really important to keep in mind because that's why we are doing this 6 feet distance. That's why we are doing this social distancing, that we are not getting close to each other because there's this pre-symptomatic stage is really important for transmission, even though when we don't know that we might be infected, but the virus is still in our nasal, in our upper respiratory system, and when we sneeze or if we speak, these droplets can contain those viruses. So it's very important to wear masks, stay 6 feet away, and also to be to follow the guidelines of stay at home. Again, okay, let's move on to replication of viral genome. Okay, once the virus entered into the cell, what happens next? One of the really important points that I want you to take home is importance of these RNA-dependent RNA polymerases. These RNA-dependent RNA polymerases, these are the polymerase of the virus. These are the ones that are helping it replicate. And this RDRP is the central component of coronavirus genome replication and transcription machinery, because this is the enzyme. Okay. This coronavirus RDRP, it's NSP12. This NSP12 and two other cofactor, NSP7 and 8, are the most important structural elements, most important polymerases for this are the important part of the transcription machinery. And these are great target for antiviral drugs as well. For example, you probably heard about remdesivir. This is an antiviral drug, a broad-scale antiviral drug that has been now is on the forefront. They're looking at how remdesivir might be useful for people who are already infected, their infection level is high, and whether it can be stopped, or the viral replication can be stopped by using remdesivir or not. So we will talk about these later. So knowing the structure of RDRP is really important to design new chemical compounds that could inhibit its function, new and better than remdesivir compounds. So here, this is the structure of this RDRP. Now, again, another thing to think about here is all this work started in January, February, and you can see that there's just enormous amount of research has been done already. This is just amazing amount of data there's already published. One of the things that is really important about this RDRP in the SARS CoV-2 is that there is a beta hairpin on the N-terminus of this RDRP, which is unusual, which is unique to this virus. This hasn't been seen in other RDRPs yet. These models have been made to understand how the antiviral drug might bind to RDRP of this particular virus, and also what's the function of this beta as well. So there are more studies going on in that. So here, this is a larger image. So this is the structure of CoV-2 RDRP. Like most polymerases, they have these different domains, finger domain, palm domain, and thumb domain. Remember, we talked about the polymerases when we were talking about these polymerases are kind of like right-hand structure. So they have this thumb, palm, that's where most of the polymerase activity goes on, and fingers that will check, finger and thumbs will check whether it's correct or not. So one of the really important thing about RDRP is these are not very foolproof enzymes. So they are not, they make more error than any of the polymerases that we talked about, the DNA or RNA polymerase in prokaryotes and eukaryotes do. So these RDRP are essential polymerase enzyme for RNA viruses. These are essential. These are encoded in the genome of all RNA-containing viruses that has no DNA stage, which is pretty much all RNA viruses. So either a negative strand or positive strand RNA virus, they all have RDRPs as their main polymerase. These polymerase catalyze synthesis of RNA strand that is complementary to a given RNA template. So they don't use DNA template, they use RNA template to make an RNA strand. The initiation step of RNA synthesis begin at or near the 3' end. So here, this is the 3' end of the RNA. And it's used, it could be two ways. It can be a primer independent, that means de novo, or it can be primer dependent, but this primer is already present in the genome. It is a viral protein genome linked primer, VPG, which is the viral genome codes for this primer if it is a primer dependent mechanism. Unlike other polymerases, the RDRP have high mutation rates, which is really important to generate the variability in RNA viruses, because that's the reason you hear this as a lot, because these RNA viruses, they mutate frequently, and because they mutate frequently, they can get over these any drugs that we are making. And this is also the reason that you need a flu vaccine every year, because every year there will be a new strain of flu virus, because they mutate pretty rapidly. So within a year, the strain will be different, and then that's why you need a new vaccine for flu. And you might remember a couple of years ago there was a very bad flu year, and the reason for that was the flu vaccine that was made was already there, that we were giving to everybody, was for a different strain, but the strain that year was different from the vaccine. So the vaccine didn't really help, because this strain wasn't targeted by that vaccine. So this RDRP brings this high mutation rates in the genome, and this is important. This is that benefits the virus. This is their evolutionary strategy. RDRP can also create recombination, and remember we talked about earlier that how this COVID-19 causing virus, this SARS CoV-2 might be a recombination. Might have been recombined from two different viruses, a pangolin virus and a bat virus. So this RDRP is also important for that, at least for poliovirus. But it appears that in SARS-CoV-2 there's an additional enzyme. We will talk about that in next slide. That might be more important for recombination. So here, this is again, it shows the same thing. These are the spike proteins on top. These attachment and entry. Spike protein binds to ACE2 receptors. It goes in. This is uncoating. Those proteases will cut this here. This because this cuts this the spike protein bind to it. This will allow this genomic RNA, this RNA, positive strand RNA, to be released. This is the RNA that has been released now. Now these ORF1 and ORF2 will be translated. Those will make the RNA-dependent RNA polymerase, or RDRP. This RDRP will then uses the host cell machinery and will to carry out this transcription or replication, whatever we call it, because it's RNA, so it will probably transcription will be the better term here. So this uses here 5' negative. Then this will translate these different proteins as spike protein, nuclear protein, envelope protein, membrane protein. All these will be translated, as well as this the genome is transcribed as well. So here goes to endoplasmic reticulum, Golgi body. These virion gets assembled along with the RNA inside and the proteins outside, and then exocytosis. Here happened the viral release. Okay. As I mentioned, that recombination of CoV RNA might require this viral, or requires the viral NSP14. This is a preprint. This is not a peer-reviewed article yet, but there's a lot of research right now on SARS-CoV-2 as in form of preprints on either bioRxiv or medRxiv. It's coming from either clinical side or more basic research. A lot of people, they put their findings, publish their findings in these databases. In these are called preprints because these aren't yet published formally. So these the coronavirus proofreading exoribonuclease. This is NSP14. Remember, the RDRP in coronavirus is called NSP12. So this is not the RDRP, but it's a different enzyme. It's an exoribonuclease. It's a proofreading enzyme that can lead to extensive viral recombinations. So these viral recombinations within the viral genome to bring the more diversity in the genome of within an organism, or the recombination of different different viruses as well. These authors also showed that SARS-CoV-2 and MERS-CoV, they undergo extensive RNA recombination in cell culture. That doesn't really mean that it will happen in the cells as well, because cell culture is pretty different in vitro system compared to in vivo system. But there are possibility that if it happens in cell culture, it might be happening in the cells as well. So again, it brings the differences, the diversity in the genome of the virus by doing the recombination. Remember, when we talked about recombination, that's the major function of recombination is to bring the to diversity in the genome. Genome itself is stable. Genome itself is the same, but the sequences of the gene of the genome might change, whether it's inverse recombination or the recombination has moved from for example, in eukaryotes, it might be a change of the arm of a chromosome. So all these different ways it can bring the diversity. Okay. I wanted to make sure to bring your attention to this, to hear that this NSP14, this exoribonuclease, is a determinant of both high fidelity CoV replication and recombination. And again, it also represents an attractive target for virus inhibition and attenuation. You will probably say that we just said in previous slide that these are not very high fidelity. The replication is not very high fidelity in viruses, in RNA viruses, because that's beneficial for the virus. But why this fidelity is? We're talking about fidelity now. So even if there is an enzyme that's checking, proofreading the genome of these RNA viruses, but still there are plenty of mutations that are caused during transcription, because one thing is the transcription is much faster. Other thing is RDRP, the polymerase is not checking. So everything that is checking, the fidelity is only through this other proofreading exoribonuclease. Okay, so now this is more about the biology of SARS-CoV-2 that we talked about so far. Now I will move a little bit more into where we are in terms of diagnosis, where we are in terms of treatment of SARS-CoV-2. Well, let's talk about a little bit about symptoms that are associated with SARS-CoV-2 infection. So, like any viral infection, there are different stages of infection. So, stage 1 is the early infection, or you can call it asymptomatic infection as well, where this is the viral response phase. The virus has entered into the cell. Virus is now replicating, and then the body is our own body is trying to respond to that virus, that foreign material. So our immune system is at this point was working to keep the viral load low. Stage 2 is the pulmonary phase, where it's actually start affecting the lungs, the respiration, so it leads to the shortness of breath or hypoxia. So the clinical symptoms in stage 1 are mild. So these are maybe a mild fever, dry cough, diarrhea, headache. So those the very first symptoms, very similar to flu symptoms. So these are the early infection symptoms. Then in stage 2, in pulmonary phase, shortness of breath, that's where most of the time will be the infected individual will go to see a physician, they might need a ventilator at this point. Stage 3 is hyperinflammation phase. That's where the host inflammatory response is very high, and that can lead to a shock or cardiac failure if not treated at this point. What are the potential therapies for SARS-CoV-2? And we will talk about these. These are all potential. None of these has been really we know much that if it's going to work or not so far, because we are still in very early stages. Remdesivir sounds because it's a broad spectrum antiviral. The other broad spectrum antiviral also appear to be helping to be working against the virus. Chloroquine, hydroxychloroquine, these are antiparasite, these are they also have viral, these are also antiviral-ish. So these, how good these are to block the viral entry or viral multiplication, the transcription is it's still we still don't know. The studies haven't been conclusive yet. There's only 2 studies so far that I'm aware of. The very first one that came from Seattle, and then they said that this was working, and then there was another one that showed it's working. However, all those studies were in the cells, in an in vitro system. So cells is very different from the person in the body. So again, unless we know more about how these work, we probably won't be using these. Another way, another therapy that's now is some of for the highly infected people, those people that are at very, very high risk of death, has been used is convalescent plasma transfusions. And convalescent plasma transfusion means people who have been infected, they have developed antibodies against the virus, so their plasma has these antibodies. So injecting those antibodies into the patients that they don't have enough antibodies might help fight back the infection as well. So these are different than IL-6 inhibitors, because it's as I showed in earlier slides that the ACE2 is the increase, that this binding of spike with ACE2 is increased when IL-6 interferon 6 levels are high. So those inhibitors might be helpful as well. So right now there is no drug or no therapeutics that are approved by FDA for use, prevent or treat COVID-19. Everything that we can do right now is prevention of infection. So stay at home, control measures, wear masks, wash your hands, all those kind of things. Supportive care, include supplemental oxygen, mechanical ventilator when needed, so that when you go in second phase, that's all is there. However, there are a lot of work going on, and this is just shows you just a little screenshot from NIH website. So this is NIH is working on the funding research for a lot of different research to figure out if there is any drugs, any existing drugs that can be repurposed for treatment of COVID-19. So you can see that there are a lot of different drugs are here that been in our convalescent plasma therapy. More drugs, hydroxychloroquine, all of these. So these are there are a lot of research going on at the moment, but still we don't have any drugs. So if if you're talking to your family, and if they're because you probably are the experts now because you're biologists, and if they ask you about these drugs, so the answer right now is we don't know much. We don't have enough information. So what's the projected timeline for treatment and prevention? This is one of the questions that I get a lot from my family. What do I think when there will be a treatment of COVID-19? Well, that's really hard to say, because any good vaccine takes time. It takes a long time. The projected timeline of 12 to 18 months, it's still towards the shorter end. I won't be surprised if it takes longer than a year to come up with a good vaccine. And I will tell you why, the reason for that. So probably the more short term is repurposing drugs and the antibodies as well. So repurposing drugs, probably if within a couple of months we should be able to know whether these drugs work or not, when we will have more people, more data to prove whether it's working or not. Antibodies, we talked about, as that will probably take another 5 to 6 months at least. Not yet, because those people, we still don't know how many people have been infected. So that's another thing. Because the tests are so few, it's really hard to know how many people are infected. And because there is no seropositive test applied so far, there was just one study from Stanford in California, which was actually was not a very good study. So that's the seropositive tests need to be carried out in order to know how whether there is a big population that was infected and they have recovered, so we can start using those antibody therapy. Vaccine takes the longest time to develop because they have to go through a lot of these different phases. So GMP, that's really just letting FDA know that this is the vaccine that we are going to be using. This is also, that's where all the controls and everything need to be shown to FDA that it works. Then it goes in phase 1, and then it goes in phase 2 and 3. So before it goes in phase 2, phase 3, before it's the efficacy of vaccine is known, the side effect of vaccine is known, it cannot be used. There are some of the vaccines are already going, and one from the Oxford is going in phase 1 and phase 2 together. So that's probably, if it works, it will be the first vaccine to come out, and that will be great. Okay, so repurposed drugs. So we already talked about remdesivir. Remdesivir is a broad-spectrum antiviral drug. This was originally designed to target Ebola. This seems to be pretty effective at fighting this COVID-19 as well in isolated cells. So that's the trick here. So everything so far is done in isolated cells. But there's better proof that remdesivir might work with this COVID-19 infection or SARS CoV-2 virus, because and I will show you how it binds to the RDRP, the polymerase, and this binding is pretty good, and it should be able to block the RDRP the enzyme. And if there is no polymerase, that means the infection will not be able to move on. So you might have noticed here that there are the drug targets different levels of infection. So this remdesivir is targeting the infection that's already there. It's not affecting the entry of virus into the cell, but what it's affecting is the transcription of the cell of the virus into the cell. Okay. Chloroquine, hydroxychloroquine, these are malaria drugs. So they are also used in autoimmune diseases. They appear to be effective against SARS CoV-2 in one study in vitro, but really there is no conclusive evidence that chloroquine or hydroxychloroquine does affect SARS CoV-2. APN-01, it's in China, the trial is going to start. This is an ACE inhibitor. Potentially, that's what APN-01 is. So it inhibits the ACE2. So because it inhibits the ACE2, that means it's can affect the entry of virus into the cell. So this is a different target. Remember, the spike proteins have to bind to this receptor. So if it's not binding to that receptor, it's not able to bind to that receptor, then it will not be able to infect the cell. Other broad-spectrum antiviral drugs are also being now other scientists are looking at those. So this is the model that the scientists came up with, incorporation model of remdesivir in SARS-CoV-2, NSP12, which is an RDRP. So this is RDRP of SARS-CoV. This is remdesivir. So remdesivir appeared to be tightly bind to this RDRP where the cofactors will bind. So remember, there are two cofactors along with this RDRP that are required for the activity of this enzyme. So this remdesivir inhibits that binding. Vaccines. There are several approaches that has been used right now: whole virus vaccine, recombinant protein subunit vaccine, antibody vaccine, nucleic acid vaccine, and you're probably aware of all these already. Whole virus vaccine is where you use the whole virus. So this virus is attenuated, weakened, or dead, and that's injected. That will trigger the immune response. And the body. Recombinant protein subunit vaccine is, instead of using the whole virus, attenuated virus, you use a recombinant protein subunit of the vaccine for development of vaccine, recombinant protein subunit of the virus. The risk of infection in people who receive them is lower in recombinant protein compared to whole virus, so that's why these are a little more better, I would say, approved by the public. Antibody vaccine is a vaccine that's using antibody from the other SARS outbreak that began in 2002. So this, because SARS and SARS CoV-2 are quite similar, so there's another idea that the antibodies against the SARS might work for SARS CoV-2, but that's seems a little far-fetched. Because we just saw that SARS and SARS CoV-2 are quite different. They're similar, but they're also very different because they have different binding sites to ACE2 receptors. They also, the SARS CoV-2 uses the multi-basic sites, not the SARS CoV-1. So antibody against these viruses will be probably pretty different as well. Nucleic acid vaccine is, instead of using the protein, you're using DNA or RNA. Inject DNA or RNA of the virus into the live host. And then these DNA or RNA in this case, this will code for proteins. Those proteins will then initiate an immune response. And that's another way. There is no nucleic acid vaccine. You probably have heard of RNA vaccine. There was a really nice program on NPR 2 weeks ago, I think, where they described this RNA vaccine in quite detail. I will see if I can find that clip. I'll send it to you, or you can just Google it. RNA vaccines or DNA vaccines, these nucleic acid vaccines haven't been used in humans yet, so this will be a really new experiment if this works. I already talked about this, that ACE2 or TMP-RSS2 inhibitors are a pretty good strategies to inhibit the entry of virus into the cells. How long it will take to design a vaccine? Well, that's what we know, what our knowledge and experience with other viruses. A vaccine is probably not going to be possible during the current outbreak, so it will be good to avoid the next peak of this infection, but probably not this year. This is going to go into 2021. And why it takes so long to develop a vaccine that is safe and effective? Because the first thing is, step one is the first you find, once you have a potential vaccine, you have to send it to the FDA to get approval for manufacturing of an animal testing of this vaccine. In step two, that's where your phase 1, phase 2, phase 3 studies start. Phase 1 evaluates the vaccine safety and ability to generate an immune response in small group of people. Many vaccines pass through phase 1 and then they just don't work in phase 2 and 3, because phase 2 and 3 that's where you use a lot more people, right doses, because the phase 1 is usually pretty high dose. You just want to see whether it works or not. In phase 2, it has to be more effective, and it has to be safe as well, and to determine the right dose. Phase 3 is where you look for the effectiveness of the drug or the vaccine, whether it's really working or not. So what do we have right now is, we don't really have anything to avoid COVID-19 infection. Everything that we have is, once you are infected, these drugs might help, but before that, all we can do is social distancing that we have been doing. We implemented the social distancing measure. A lot of people there has been a lot of debate going on around social distancing, that social distancing isn't really doing anything, it's not working, people are still getting infected. But one thing to keep in mind is that social distancing was never to not get infected, that people will still get infected, but it is more to lower and delay the peak, so to not overwhelm our medical system. So if there's an uncontrolled transmission, this would have been this is just a model. In an uncontrolled transmission, this will be the peak here early on, and if this peak goes so high early on, that means our medical facilities, our healthcare system does not have the capacity to take care of that many people. But if we use social distancing, this peak will come, but it will come later. Peak will be smaller, infection will be over time, so the healthcare system will still be able to treat all these people. So we will still have enough ICUs, we will still have enough ER visits capability if we do this. So this is the model. Again, let's just keep doing what we should do, hand washing, working from home as long as we can, as much as we can, limiting large gatherings. So as the mandate is no more than 5 people, minimizing travel. Probably nobody's traveling right now, but even in the coming months, so it's not short term, as you might have seen through all this presentation, that we don't have anything to avoid infection. We don't have vaccines, we don't have drugs to treat us. So really what we need to think about is, and it will take about a year from now for a vaccine to be developed. So we have not just the short term, but all these things that the control measures that we are taking place, that we are working on now, need to be long term, at least for a year or two. So what are the key questions that the SARS related to SARS CoV-2 immunity that you're probably thinking about, or you're probably asking is, okay, if there are people are infected, will there be what's the correlates of protection? Is there really the seroprevalence in population will help? Antigenic diversity and cross reactivity, is that a thing, right? So what's the kinetics of immune response to infection? So all these questions, these are all valid questions that we need to ask as we open up the economy, as we open the country, we open the state. So we have to think about all these questions. So what's really important is, you might have heard a lot of debate about that we don't have enough tests. Yes, we don't have enough tests, but those RNA tests, nucleic acid tests, aren't really going to tell us much at this point. What we really need to invest into is a seropositive test to know how many people were actually infected, how many people have antibody developed, and how many people have enough antibody titers that they probably will be good for maybe 6 months to a year. We don't know how long the immune response, the immunity to this virus lasts, because this is novel virus to humans. There haven't been cases, so we don't know. But if we go by immunity of flu or SARS, the reason we get every year a new flu shot is because the immunity is only for a year or so, and the new strains come and in that time period. So this is going to be same thing with this, probably the SARS CoV-2 as well. So do we have to keep getting the vaccination? How long are we safe? So if I had an infection and I developed antibody, and I go, if I'm good to go back to work, how long will that immunity last? So these are the questions that we don't know, that we're still working on. So seropositive testing. Seropositive testing, it's most of you are aware of seropositive testing, right? This is very similar to a pregnancy test, where you're looking at the antibody response. This little this stick has antigens. These antigens will bind to the antibody if we have antibody in the saliva or whatever is being used. So if you can use it for saliva, so it's you put the saliva in a tube, you put this little the tube, the little test on that saliva, and that the antibody will move. And if there is if somebody's positive to those antibody, if they have enough antibody developed, this will give a double band versus a single band. So test line versus a control line. So this is the seropositive testing, which is really going to be important if we are going to open the economy. So final note, I will leave you at this. Do not, under any circumstances, I know you're not going to, but tell everybody that do not spray or introduce bleach or any other disinfectant into your body. Another thing is, please do not buy into the claims that chloroquine is a magic miracle drug, because there aren't any studies suggesting that it is. So tell people not to buy chloroquine, because people have been buying. And with that, just stay safe, stay home, wash your hands, and use mask when you go outside. Stay safe. Thank you.